to my poster talk. My name is Nikolaus Fortelny, and I'm going to be talking about the computational analysis for functional screens in hematopoietic development. I'm a group leader at the Paris Lodron University of Salzburg, and we've been working on this together with members of the University of Navarra and the University of Cambridge. This project is currently in preparation for publication in Nature Genetics, meaning it's basically accepted and should come out soon. In this project, we analyzed single-cell CRISPR screens in a developing system, specifically hematopoiesis. Our screens were focused on chromatin factors, so CF here, which affect developing trajectories. And while transcription factors are highly studied in development, chromatin factors are often not as easy to study. So this project is based on experimental single-cell data where mice, GFP mice were taken, and the bone marrow from these cells, these mice was taken, and the bone marrow was transformed with viruses harboring guide RNAs for different chromatin factors. The stem cells or LSK cells from these mice were then transfused into mice with irradiated bone marrow, which were not Cas9 but wild type, so that the cells could be seen. And then after 14 days, these cells performed hematopoietic development in those mice. After 14 days, they were sorted out. The hematopoietic cells were sorted out and underwent single-cell sequencing. And then we analyzed the data to understand the effects of these chromatin factors on hematopoietic development. To first visualize the data, we performed UMAP analysis, and you can see here a clustering of the individual cells. And I would like to point out some major branches here. So on the top left, you see the erythroid branch with the progenitors here at the bottom. We also have some megakaryocytes. Here are the stem cells. Here is the lymphoid lineage, just represented by B cells, so this was underrepresented here. And on this side, we have the myeloid branch with the myeloid progenitors and granulocytes and also monocytes. Now looking at these cells, some cells or many cells were non-targeting controls, so they didn't have a knockout, and then multiple cells had a knockout of certain chromatin factors, and you can see that these create some very interesting patterns. Sometimes they enrich in a whole lineage here, sometimes they just create very specific clusters, and sometimes within the lineage they drive it to the end, for example, the CHD4. So how to analyze this statistically to get relevant information? We first used the clusters to understand whether certain chromatin factors enrich in certain clusters. So this is what these plots show. Specifically, this one has some broad clusters from the previous plot, and then dots which show whether cells are enriched or depleted in a certain cluster. And you can see our chromatin factors here. Blue means depleted, red means enriched, and you can see very clear patterns here, as similar to what we saw in the UMAP. We also asked some more specific questions. For example, are certain chromatin factors enriched in early myeloid versus early erythroid development by just taking the early clusters from this UMAP, or just myeloid versus erythroid development in general, or even within the myeloid branch, the monocyte versus the granulocyte enrichment. So these enabled us to ask very specific questions of the developmental effects of these guys. We were also able to assess viability by comparing to the reads in the library before transforming the cells. So this enabled us a number of analyses, but going too detailed with those clusters was almost impossible, because if you have 20 clusters here, it is hard to interpret or even visualize them. So we wanted to find some continuous way of analyzing these screens, and what we turned to in the end was trajectory analysis. So plotting all of these cells on trajectory tools. So there are a number of good trajectory tools in the single cell field, and measuring an erythroid, a lymphoid, and a myeloid trajectory. And then what we did was we obtained pseudo-time values from them, so these quantify how far cells are in those trajectories. And then we could compare those between the non-targeting controls and the different guides and knockouts. And you can see here that certain guides really shift the development, either by retaining cells in the early stages of a certain trajectory or pushing them to the late stages. This enabled a more continuous and fine-grained analysis. So with this, I would like to close. I want to thank the people who contributed. On the right here, this is my lab in Salzburg. And on the left are collaborators. David Lara Astiaso, he really pushed this project. He performed most of the experiments and also coordinated a big part of the experiments. Ainoa here helped with some of these experiments. Nisha performed some analysis of leukemic cells, which we haven't really shown in this presentation. Hulen performed analysis of epigenomic data, and Brian is the PI from Cambridge. Yes, as I mentioned, this paper is currently accepted in Nature Genetics and should hopefully be published soon. Maybe by the time you see this talk. Thank you for attention and goodbye.